I prepared some data from a case study that we did on the protein MYC. So MYC is an intrinsically disordered transcription factor complex. So it's actually two proteins, MYC and MAX, that form a heterodimer. And when this dimer has formed, it can bind to a DNA, a specific DNA sequence. It's called the EBOX DNA. And by this, it can regulate gene expression. And the goal in drug discovery, for example, would be to find inhibitors, so small molecules, that can first prevent this dimerization from happening, and by this also prevent that the DNA binds to MYC. So when you have to deal with the system MYC and you want to measure binding affinities to small molecules, there are a couple of must-haves that you should have. So first of all, it's a transcription factor, which means that MYC does not have a biochemical activity. So you cannot use biochemical assays to measure binding. You really need a biophysical approach if you want KDs for this protein. Also, as we've seen on the previous slide, there's not only inhibitors binding to MYC, but there's also this interaction with the EBOX DNA. So it's a bit more complex system, and you want to have an assay that is flexible enough to get all these different interactions analyzed. So not only small molecule binding, but also maybe displacement from the DNA. And finally, you should have a method which does not perturb the conformational equilibrium. So this is really important for IDPs. Sometimes when you immobilize them, for example, on a surface, this can really affect their conformation or the space of conformations that they can take. And then maybe they will be in a different conformation on a surface, and you actually find wrong inhibitors that bind to that conformation, but not anymore to the conformation that it has in solution. So it's really recommended that you study this protein in an in-solution assay. And with Dianthus, you can get all these roadblocks basically overcome. So first of all, as you've seen in previous talks, we have two biophysical modalities in the Dianthus, that is Spectral Shift and Trick, that can measure binding affinities. It also allows you to label your molecules in a very flexible way. We have different labeling kits, and you can think of labeling different molecules. I will show you this on the next slide. So you can study both interactions. And finally, also, it's a fluorescence-based method. So it means we are working in solution using a very sensitive readout of fluorescence. And this also allows us to work at very low concentrations. And this is another thing that you should keep in mind if you have an IDP. If you have it at very high concentrations, these proteins really tend to aggregate, and this can mess your assay up. So if you can work at very low concentrations, it's very recommended, and this is possible with the Dianthus. So let me show you how we tackled this system. So we started with the protein. We got MYC as a dimer already formed. So the question was not to label either MYC or MAX, but the dimer together. And then we thought, how can we label it? So first of all, you would think of different tags. Is there a his tag? Is there maybe a free cysteine? We decided for NHS labeling. So this is the most general way to label a protein. You randomly label lysines on the protein. And this then allowed us to measure binding of inhibitors to MYC. And then we also thought of a different assay where the DNA comes into the game. And there we thought if we label the DNA, we can also study first if MYC binds to the DNA, and then also if there are small molecules that can disrupt this binding and then displace the protein from the DNA. And how we did this was we used a DNA sequence that had been described in a paper. The authors had used biotinylated DNA, and we took the same sequence, and instead of the biotin, we attached a Cy5 dye, and then we got it labeled, and we could measure the interactions with that. So we started with a very simple assay in which we took the labeled EBOX DNA as a double strand, And then we just increase the concentration of McMux to see if the protein that we got, if it's active, if it's functional. And you can see a very nice dose response curve where we have 5 points in saturation. So this is always good to have. It was very tight binding, as expected, in the nanomolar range, so 5 nanomolar KD. And also you can notice this transition is very steep. This also happens because MUX itself, as a dimer, has an equilibrium. So if you actually dilute it to too low concentrations apart, and then of course it cannot bind. So that's why we're here. So once we had this established, we looked also at displacement. And the easiest to do this without any inhibitors is just to add unlabeled DNA. So we have here in the beginning the labeled DNA bound to McMux, and then we increase the concentration of unlabeled DNA. And at some point, you will achieve that the McMux jumps from the labeled to the unlabeled in the equilibrium state, and you end up with a signal that is again low. So this is here the spectral shift ratio, and it's again the same value that we had here on the left when we started the assay. So if we get down to this point, it means we have successfully displaced the McMux from the DNA. So here's how it looked with the inhibitors. We started with one, it's called 10,058 F4. It was one that was quite early described in literature. And when we added this to the labeled McMux, we could see a spectral shift. So there was a red shift of the red dye fluorescence, and the binding here indicated a KD in the range of about 20 micromolar. So all these small molecules, they have quite weak affinities. You don't see here any nanomolar binders because there's just no binding pocket on this IDP. It's rather that this molecule has to bind on a large interface on the protein, and that's always hard for these small molecules, so we don't get really tight binding. In the displacement assay on the right that you can see here, we also started with the labeled DNA and the McMux, and then increased the concentration of the compound. And as predicted, we could see that the ratio decreased, meaning we have here a proof that this small molecule is able to displace McMux from the DNA. We don't reach complete saturation because we couldn't go high enough with the concentrations, but it's definitely displacing the protein. The second molecule we looked at was called EN4. That's a newer inhibitor. It's also a covalent binder, so quite interesting. And that one similarly showed binding in the direct binding assay, again in the mid-micromolar range. But what was very cool to see was in the displacement assay, we could here, for the two highest concentration that we studied in this assay, we could see almost complete displacement. So here really the ratio value went back to the value of free DNA. So this molecule is really potent and can displace McMux, even at the second lowest concentration, almost completely. And then we looked at the third one, the last one I want to show you. And that was quite interesting because this molecule KJ-pure9 had been described in literature as a binder. But then later there was a paper where SPR was used, and the authors could not confirm binding, and they didn't have an explanation. So they stated they simply couldn't see any binding of this molecule. So when we used it, we initially thought, it's also binding. We also saw a similar dose response in the direct assay. But in the displacement assay, we did not see this decrease of the curve. So before, the two curves, they also went down. But here, the signal increased further. And this probably means that the labeled DNA keeps being bound to McMux, and then also the molecule is binding. And probably everything is just getting together and maybe aggregating, but we don't have the displacement. So this molecule is not doing what it is supposed to do. And we also looked at the well scans. And as you've heard in the previous talk, sometimes the well scans, usually they should look nice, like a cylinder, like a head. but sometimes they have this bumpy traces, and this means usually that you have aggregation. So what we believe here is that this compound binds to MYC, but MYC stays on the DNA and everything just aggregates, and it's an aggregator. So it's really cool that we can identify this molecule using our technique. And yeah, it's not a desired behavior. So I hope I could show you a bit what you can do with Dianthus when you work with IDPs, challenging intrinsically disordered proteins. I think the strengths are that you have this biophysical method at hand using fluorescence, and you can work in solution, which is always good for these IDPs. It's a very flexible assay design that you have at hand. So you can set up assays like displacement assays. Also, if you have, for example, a small molecule that is fluorescently labeled, you can use it as a tracer. So there are many ways how to design the assays. All the KDs we got were in the expected range. So literature data also was always this high micromolar range for these MYC binders. And it was very nice to see the behavior of this KJ-PIR9 compound that we maybe could clear up how it's working on this protein. And finally, also covalent inhibitors are not a problem. So there's no worry that you bind something irreversibly to, for example, a surface, but you can always just use new wells. So we are working in the micro well plates, and every experiment is just a new row of wells. So there's no need to regenerate.